Hello, everyone. Welcome to today's live broadcast, Tough Targets, Simple Genotyping, using HRM as a direct approach in research applications. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Canon BioMedical. Canon BioMedical, a wholly owned subsidiary of Canon USA Incorporated, is focused on empowering the biomedical research and healthcare communities by developing, manufacturing, and marketing innovative technologies and solutions. Before we begin, there are a few instructions. We want to hear from you during this interactive broadcast. So please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we will follow up if we don't have time to answer all. If you want a better look, you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see the presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. This is also an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now, let's get right to today's presenter. We are proud to welcome Dr. Brian McNally. Brian has 15 years of practical experience at the laboratory bench, and his work has been published in peer-reviewed journals and presented at international conferences. In 2008, he transitioned to industry to commercialize new biomedical products, including assays, reagents, and software. Brian has been with Canon BioMedical since its inception last year. He is passionate about partnering with life scientists to develop the next wave of biomedical solutions. Dr. McNally, the presentation is all yours.
Thanks very much. I certainly appreciate the kind introduction. So we're going to go ahead and get started today on a topic, tough targets and simple genotyping, and as the sort of obligatory slide that I have to put forward to make my colleagues in the legal department happy. All of the Canon BioMedical products that I'll show you in the presentation today are intended for research use only. They are not for use in diagnostic procedures. Those of you that relish looking at licensing information and product-specific disclaimers, I encourage you to download our user guides at canonbiomedical.com, or certainly put a request in the Q&A section, and we'll go ahead and send that to you right away. The Canon BioMedical products that are going to be shown today are going to be available to you today directly from Canon BioMedical in the United States. For those of you that are in the United Kingdom and Ireland, they are available through a distributor, LabTech, which would offer those products. Those of you that are in other countries, we're working very hard on getting our products to become available in those. So if you are coming from another country and you have interest in these products, please just indicate that in the Q&A box, and we'll be happy to work with getting them available to you as quickly as possible. And finally, the Novallele is a trademark of Canon BioMedical Incorporated. So with that out of the way, the lawyers are all happy. A lot of you might still be scratching your head. Canon at Clinical Diagnostics and Research. And so we get a lot of different questions about this. Are you part of the camera company? Are you part of the printer company? And the answer is yes, they are our parent organization. And then in this particular case, we're a new wholly owned subsidiary of that. And so the next thought that then often gets asked of us is, well, you must have something to do with microscopes or imaging, and I think I'm going to surprise you in the upcoming slides. But with that in mind, I'd like to ask all of you, how many of you have heard of Canon BioMedical before? So we're going to launch a poll right now. And if during that polling, you would go ahead and please just let us know, have you heard of Canon BioMedical before, yes or no? It just gives me a good sense of the familiarity the audience has with us as an organization. So I'll give you a few seconds to go ahead and click on those responses. certainly really appreciate you using the polls during the course of the presentation today. It's extremely helpful. All right, well, thanks again. So I'd like to tell you a little bit about the history of Canon and where Canon BioMedical is coming from. And I'm going to begin this with a question that's sort of open to, everyone's guessing. Has anybody, does anybody recognize what this X-ray is? And I've gotten some very clever answers from giving this presentation before. I had a respondent answer me that, yes, this is an X-ray. When I told the next audience that I had previously been told that this is an X-ray, the next audience responded, this is a chest X-ray. So both of those statements are true. But to help everybody out, this is actually a X-ray showing a patient with tuberculosis. And in the 1930s, this was considered Japan's national disease. It was a rampant disease across the country, affected well over 1 million individuals, making them very, very sick. And so in the late 1800s, there were direct X-ray cameras, and you can see an image that was taken from a popular electricity magazine, and you can see there a man holding what would be considered a direct X-ray in front of this woman looking for either tuberculosis or some other growth or something like this. Well, these large format X-rays were at the time in the 30s very, very expensive. And it really needed sort of a collaborative approach to then solve this medical need so that we could had transfer from X-rays really being only used as a diagnostic tool to more of a screening tool. And so that's what happened in the late 30s. A medical doctor approached another medical doctor named Takeshi Mitarai, or Dr. Mitarai. And Dr. Mitarai was one of the founders of Canon, and he was the first Canon president and served until 1974. And this other doctor brought together an idea that allowed for an indirect X-ray imaging, which would essentially take that chest X-ray and then expose that onto 35 millimeter film. And so Canon saw this as really a way to contribute to society and positively impact the individuals within the country that were suffering from this debilitating disease. And so this collaborative approach of working with the medical community to take an advancement, innovate on it, and then deploy it broadly really was one of our first steps as a company into the world of diagnostics. And so screening in 1939 with the release of the CX-35, this indirect X-ray camera, really helped turn the tide and stem an epidemic in Japan of tuberculosis. And stepping through time, Canon has constantly interacted with the medical community to positively impact our healthcare. And so I would argue that in the 60s, there was the deployment of these indirect X-ray cameras out into the field on these buses. So you can see in the image in the right-hand side there's a little bus. That bus is carrying the upgraded version of these X-ray cameras. And this bus is located in this picture in Nepal, which was also fighting at that time tuberculosis. And I would argue that this was Canon BioMedical, or Canon's rather first steps into the idea of point-of-need testing, is through putting a diagnostic device on wheels and deploying it to remote locations around the world. Those advancements continued. In 1967, Canon joined forces with the National Cancer Center in Tokyo, now not to focus on infectious disease, but rather to focus now on cancer, and in this particular case worked on developing special imaging technology that was then used to screen for stomach cancer. And then moving now into current day, in 1997, Canon invented the first digital radiography solution. I've shown here in a picture on the right-hand side the RAD Pro Elite, which is now a current model that's used around the world for digital radiography solutions. So we've taken sort of a film-based approach, a collaboration, used it originally to screen for tuberculosis, then went on to point-of-need testing with putting that technology onto a bus, broadened the types of diagnosis we could use through imaging with cancer detection, and then, of course, now took a step into the digital world with a digital radiography solution. And so at the heart of all of this is really, sort of an imaging a macro sort of based diagnosis of medical conditions. But what we can see in the biomedical research community is that we're moving beyond these whole body imaging type solutions into much more cellular and molecular type solutions. And so that's where the field is going. And so Canon, again, wants to continue in that tradition of collaboration with seeing how that we can then help with the world of cellular and molecular. We began that with Canon BioMedical supporting two partnerships. The first one shown here on the left with T2 Biosystems. T2 Biosystems has an innovative solution coming out of Massachusetts, a fully automated sample-to-answer molecular diagnostics platform that first focused with an FDA clearance in 2014 on a Candida fungal panel. And Canon BioMedical is working together now with T2 Biosystems to collaborate on a Lyme disease test using their platform. And then another collaboration that we've begun is with a Canadian company, Spartan Biosciences. They have a point-of-need device that's a fully automated sample-to-answer solution doing a particular genotyping test that impacts the use of Plavix. And this has been cleared for a point-of-care designation by Health Canada. And so in each of these cases, Canon is bringing its expertise in things like IP strategy and core technologies, manufacturing, technical support to these organizations to help promote their solution. And that really represented our first step in the field of molecular testing. So what we want to now begin to talk about is really what Canon's products, Canon BioMedical's products are and solutions that are specifically geared towards you. And we're going to begin with a discussion of genotyping, and then I'm going to talk about tough targets in genotyping and how to make that process simple with a different assay design approach, and then I'll summarize and take questions at the end. So before I get too far along, I would like to talk about sort of tough targets and simple genotyping. I'd like to launch another poll question, which is how many of you are currently genotyping samples today? So I'm going to launch that poll now. And again, the question is, do you or anyone in your laboratory currently genotype human samples? And so I'm going to leave this poll up for a little while, and then I'm going to have a follow-on poll that's going to address the results of this one. So I'll give you all a few minutes here, or a few seconds here, to go ahead and click yes or no again. Do you or your laboratory currently genotype human samples? And now with the results of those, for those of you that answered yes, that you're doing that or your laboratory is doing it, which of the technologies listed here are you currently using to do that? Are you using next-generation sequencing, Sanger sequencing, mass spectrometry, PCR-based methodology, or something that's not listed above? Okay. This is great. I really appreciate everybody taking the time to answer the questions with regards to your genotyping and the current methods that you use for genotyping. And I wanted to talk about these in the upcoming slides a little bit more so that everybody could understand sort of our approach to genotyping. So I think we're going to go ahead and close those polls. All right. So in slide 16, we're going to go ahead and start talking about the different technologies that are used for genotyping. I've listed essentially the same ones that were in the poll. Next generation sequencing there on the left-hand side, very sort of unbiased approach, looking at whole exome, whole genome data. Then there, of course, there's Sanger sequencing, a very classic approach for looking at more targeted regions of the genome. Mass spectrometry is being used, of course, to identify variants. And then there's a couple of different methodologies that can be used with PCR. One would be hydrolysis probe-based PCR. This is commonly known as TACMAN-based PCR. There are other fluorescent probe-labeled or fluorescent-labeled probe-based PCR methodologies. And then there's high-resolution melting analysis, which is a method that we're going to talk about more today. And when you consider the challenges that are in front of you as genotypers, really there's a number of questions that I think everyone should be asking themselves beforehand. And it's really sort of agnostic of particular company solutions, but really to help guide you, Canon recommends you consider these questions. First, are you trying to discover new mutations? Are you looking for the unknown? Or are you trying to test a very specific individual mutation? So if you're really sort of discovery, you may be looking at, I want to look at any mutations within the entire genome, the exome, a particular panel of genes, even a particular gene, or even within a particular region of a gene. But that's contrasted very sharply by the notion of testing, which is I would like to test for the presence of this specific mutation. An example would be like a live mutation in factor V for thrombophilia. Another consideration when selecting a genotyping tool is how many samples do you have? Do you have 1,000 samples? Are they all banked? Are you having a rolling submission where you get a dozen samples a day? Or is this something more modest where you have a very closed system? Maybe you only need to test a dozen samples in total. Excuse me. And sample number and the way that you're acquiring those samples can obviously impact which technology you might want to choose. Next, sample amount. What type of sample is it? Is it coming from a fixed sample? Is it a fresh blood sample? Is it isolated genomic DNA? Do you have an excessive amount of the sample? Do you have micrograms of DNA to play with? Or is this a very few, maybe even nanograms of DNA that was isolated from a fine needle biopsy, something like this? And then the last question that we recommend that you consider when selecting a genotyping technology are what are the resources that you have available to you? And what I mean by that is not only how many people do you have available and what their workload is, and not only what sort of budget and cost you're willing to put towards the genotyping effort, but even what is the technical expertise of the people that you have? Do you want them spending their time on a genotyping methodology that requires a robust level of bioinformatic analysis, or would you rather those resources be applied to other things? And these are complex considerations, and by using the intersection of these, you can sort of weigh out which particular technology is the best one for you at that given moment. And so focusing on the difference between that discovery and testing, I wanted to highlight when particular focused studies might be required. And they really break down into three areas. There's pre-screening samples, there's screening samples, and then there's the verification. What I mean by pre-screening, this is a prospective study. This is really, I want to look at samples before I take an unbiased approach. So I want to include or exclude them from my further research study. By doing that, you can then save costs and sort of limit the amount of work that you might have to do in a more laborious discovery-based experiment. Screening would be the opposite, right? This would be more of a retrospective study. And a good example of this in the basic research laboratory is when you're doing CRISPR clone generation. So if you're trying to edit the genome to introduce a particular mutation, in that particular case, you'll have a lot of clones that won't have your targeted mutation, and you're trying to pick out the individual ones that do. And so you want to screen through those very quickly. And then the last one is in verification, and this could be coming from you've done a SNP microarray or an array CGH looking for copy number, and then you want to verify the data that you got from those data sets. Or you send a sample out for sequencing. So for instance, you isolate genomic DNA, you send it out to a core facility that's done the whole exome sequencing, sends you the data back, and you want to verify that the data that you got back then indeed matches the original sample that you collected. And so by using small numbers of SNPs, you can then positively identify the data that you received back matches indeed the data for the sample that you sent out. So these are a couple of different examples that I wanted to show you where you want to maybe be taking a very targeted approach and how you would use that targeted approach to your benefit. And to do that, one of the technologies that I mentioned you could use is endpoint PCR followed by high-resolution melting analysis. High-resolution melting analysis is a classic technology. It leverages the biochemical aspects and features of DNA So at low temperatures, double DNA, of course, forms the double helix that's shown on the left-hand side of this screen. If in that solution there's a DNA intercalating dye present, then it'll bind to that double-stranded DNA and give a fluorescent signal, and so maximal signal when all of the DNA is double-stranded. As you heat that sample, as shown on the x-axis, the DNA will unwind, melt, and fall apart and form just have two single-stranded forms. That single-strand DNA then occurs at high temperature. The DNA intercalating dye comes off, and the fluorescent signal is lost. So the point at which half the fluorescent signal is lost is that temperature is then referred to as the melting temperature, or TM. Excuse me. So there are two types of assays that can be used to, that can be designed in PCR to use high-resolution melting. I've shown them here. One is a small amplicon assay that's shown on the left-hand side. So this is where you just have two primers creating the amplicon. And on the right-hand side is an unlabeled probe assay. So the small amplicon assay simply creates an amplicon, and because of the excuse me. Because of the difference in GC content of the amplicon, they then are going to melt at different temperatures, and you can see the differences in the melting curves as compared to the last slide shown here on the left. On the right-hand side is the unlabeled probe assay, and this is where maybe the short amplicons don't give you enough resolution in shape and size to be able to be easily distinguished. So what you do is you use a forward and reverse primer to generate an amplicon. And then an unlabeled probe is added to the reaction, and that unlabeled probe binds to a much smaller portion of the amplicon. And you can see that in A, in this right-hand side, that's sort of that middle graph. And that creates this hetero species, and that then can be clearly distinguished when you go under the melting conditions shown in B. So it makes it much easier to distinguish the melting profile. So these are classic ways to use HRM assays. They've been used in SNPs, somatic mutations, insertions, deletions, mitochondrial targets, copy number measurement, as well as DNA methylation. And so you can use assay solutions for answering focused questions. And what we mean by that is there are two things. One, there's the addressing of a difficult or a tough target. And then there's two, using this technology to simplify your workflow or the experimental process in your laboratory. I'm going to first focus on tough targets on the left-hand side of the screen. What I mean by that is there's difficult sequences to design assays for by other PCR methodologies, multi-target discrimination, so when SNPs are very close to one another. Insertions and deletions of all different sizes can create difficulty in analysis by PCR. Mitochondrial targets and heteroplasmic, and finally copy number. And due to time restrictions, I'm going to focus on just a couple of those. I'm going to start with copy number. I'm going to start with insertions and deletions. So this is data that we're presenting at the upcoming Association of Molecular Pathology meeting next week in Charlotte, North Carolina, where the authors from our group here at Canon, U.S. Life Sciences, Canon BioMedical, have designed assays using HRM for detecting insertions and deletions that range from a single base pair up all the way to several kilobases. And to focus on one of those, there's mutations in the HEX-A gene that are associated with Tay-Sachs disease. This is an inherited human developmental disorder that gradually destroys nerve cells. It's characterized by seizures, intellectual disability, paralysis, and loss of your eyesight and hearing. And so some of the documented genetic variations that are involved in Tay-Sachs disease and HEX-A include a very large deletion that measures a little over 7.5 kilobases. And the authors in our publication clearly identify an HRM assay that's capable of distinguishing the presence of this deletion both in a heterozygous and homozygous form. So the HRM data that I'll show throughout the presentation, you'll see the black curve represents the wild type samples. The blue sample is represented of the heterozygote, and the red sample represents the variant homozygous. And so you can see at different melting temperatures the different amplicons that are formed using this assay. This assay is commercially available, and I've noted the catalog number here in the slides for your reference later. Another difficult target, though, of course, is mitochondrial targets. And this is data that we shared at the American Society of Human Genetics meeting. The lead author, Ling Shaw, presented this. And as background knowledge, over 14 different diseases and disorders are now related to mutations that not occur in the nuclear genome, but rather in the mitochondrial genome, the DNA that's inside DNA inside the mitochondria. And they're not commercially available, fast, sensitive, and cost-effective assays for detecting these mutations. So in her presentation, she highlighted studies focused on these 5 mitochondrial DNA targets. So I've listed the genes, and those are kind of the disorders there as well, MERF, MELAS, and LHON, the associated DB SNP IDs. And then the annotation there on the right-hand side is whether it's an unlabeled probe-based assay or the short amplicon type assay. And so you can see that we use a mix of these assays. So for one of them, highlighted here for the mitochondrial myopathy, encephalopathy, lactic acidosis, and stroke-like episode disease, this is a genetically heterogeneous disorder, has a variable phenotype. But the most common mutation is this 3243A to G transition in the gene. And you can see this is an unlabeled probe assay. So you can see the very tall peak on the right-hand side of this graph there. That represents the full size amplicon. But then focusing in the next slide on just the probe amplicon heteroduplex, you can clearly see that there's a distinguishing between the wild type, the heterozygote, and the variant homozygous for this particular mutation. And this assay is also commercially available. But what's really interesting about mitochondrial targets is that the mutations that are in the mitochondrial genome don't have to be in all of the mitochondria. So each mitochondria can contain between 2 to 10 copies of the mitochondrial genome. Their range, but on average, there are several hundred copies or several hundred mitochondria in each cell. And so you can imagine that only a portion of all of those mitochondrial genomes will contain a mutation. There's a beautiful study that was done several years ago that demonstrated that the more copies of a mutation that are present, that there's a more severe phenotype that is presented. So in the case of this in the RRF, there's a differential phenotype when they first stratify the patient population by age and then assay for presence of the mutation. The more copies of the mutation that are present, the more severe the phenotype is. So Ling Shaw presented at her poster at ASHG in 2016 that by creating different admixtures that the sensitivity could be detected down to 13% of the mitochondrial genome could then contain the mutation and be detected by this assay. The last example that I wanted to share with you is another presentation that was given by the R&D group at Canon on copy number detection by using high-resolution melting. So this is looking at SMN1 copy number specifically, but to give you a sense of scale of the impact of copy number variation on human disease, there's over 500,000 variations that have been documented. They drive over 10% of all the gene expression changes that are known, and so they represent a really significant driver in human biology, diversity, and pathophysiology. Specifically, SMN1 accounts for about 95% of the spinal muscular atrophy cases. There's a deletion of the SMN1 gene that can either take it down to 1 copy or 2 or 0 copies. very difficult to design assays for because there's an SMN2 gene. The SMN2 gene is virtually identical to the SMN1 gene. And what the authors here showed using obtained clinical samples, research samples, is that using an unlabeled probe assay, excuse me, using a duplex assay, that they were able to then distinguish here between the black, red, and blue curves of the 2 copies of the gene present, 1 copy, or 0 copies being present. And the UCE curve that you see here is an ultra-conserved element in the human genome that's used really as a loading control. This assay is not currently commercially available. To summarize the tough target portion of the presentation, what I've been able to show you is data using HRM assays for the detection of insertions and deletions of varying length. I've showed you how you can use those assays to detect heteroplasmic mutations in the mitochondrial genome and how those quantification of or detection of different levels of mutation then could have an impact on phenotypic presentation. And I've also shown you how that you can use HRM now to quantify copy number using an alternative approach in PCR. And HRM-based assays make difficult genetic variations really accessible to the individual laboratory. So moving on into simple genotyping, there are really a couple of different ideas here. One would be that it's an easy protocol, and I'm going to show you that. It can deal with sample numbers, and I'm going to talk a little bit about data analysis. So hopefully most of you are familiar with the protocol for PCR, but the HRM analysis portion really doesn't complicate the protocol. You have to take your research samples of interest, you extract the DNA. We recommend that you use the same protocol or extraction method to isolate your DNA. Then you create a reaction mixture. So you mix DNA together with master mix that contains a polymerase, nucleotides, etc., as well as the assay and some water together. The master mix that we recommend with the Novallele assays is the Novallele genotyping master mix, which has been designed for rapid PCR and high-resolution melting. Finally, when you take those reaction mixtures, you pipette them into the appropriate reaction chamber, plates, tubes, and you put it into your instrument that can perform PCR followed by high-resolution melting. So in general, these are instruments that have some sort of fluorescent detection capability. A lot of times it's a real-time PCR instrument. And then finally, from the data analysis, most instruments that have high-resolution melting also have an included data analysis package. So what type of instruments am I referring to? I've just listed off some of the ones that we've tested in our own laboratories that we know that are compatible with high-resolution melting and high-resolution melting analysis. So a couple from Bio-Rad, the CFX96 and 384. From Roche, there's the LightCycler 480 and the LightCycler 2. Thermo Fisher, Life Technologies, ABI, it's the Quant Studio, is certainly compatible. From QIAGEN, the RotorGene Q has an HRM module, and from BioFire, there's a LS32 instrument. So, so that's really sort of from how easy it is to use, how easy it would be to use in your own laboratory perspective. And what I want to spend most of the rest of the time is talking really about how data analysis impacts your experimental design and how easy the data analysis is. So I'm going to contrast for a second and step back from high-resolution melting and talk about the current methodologies that are used by PCR, and those require fluorescent probes. In this particular case, what you can see is this is example data using a hydrolysis probe, a TACMAN probe. There's two colors, one for signal on the x-axis and one on the y, the FAM and hex fluorescence. And what you can see is that the experiment is then sort of balances the samples out, where the genotype that's homozygous for one allele is in red, the other is in blue, and then the heterozygous in this case is in black. And you can see that all of the signal would be in one color for the homozygote, and then when the signal is kind of mixed, then that's what really creates the heterozygote. The data analysis methodologies that draw to this really didn't require clustering. So it's when the signals are located together that you can then say, okay, the red cluster all goes together, the blue cluster goes together, the black cluster all goes together. Now, when you run experiments then on different days or even in the morning and the afternoon, you're going to get data sets. And what you can see here is in the top two figures are ones that were run same instrument, but these are on different days. Here's the sample of the data that was collected. So you can see, you'd think the red go together, you'd think the black go together, and the blue would go together. But the challenge is that then when you can actually combine these data sets into a larger analysis, you really don't get the same 3 clusters. And what you really get are 6 clusters. And that makes the data analysis very, very difficult to then combine into larger data sets. Well, why is this a challenge? And the challenge really stems from the idea that then you need enough samples to then create these different genotyped clusters. This is in contrast to high-resolution melting analysis, where you really only have to have maybe a control sample, for instance, the wild type, and then your experimental sample, and you could clearly then differentiate by just the shape and location of the peaks and individual genotypes. You don't really require clustering. It can be done on an individual sample basis, so you can sample literally one sample genotype one sample at a time. This clustering also then impacts your algorithm. And so the challenge here is if you don't have enough samples, you don't necessarily create all of these different clusters. And so I want to show you some data. This is a data set that was created using genotyping just 6 samples. You see the node plate control in the lower left-hand corner in the gray. You see a black triangle and then the 5 blue dots. And if you were to analyze this data, you'd say, well, the blue dots look like a homozygote for one allele. The black triangle probably represents the heterozygous sample. Well, if you look now at an expanded set of data where more samples were included, now what we see is, okay, we still see our gray dots, the node template control in the lower left-hand corner. Lower right-hand corner we have the blue, and then in the center we have the black dots. But notice our black triangle from the previous data set has now moved down into the blue cluster, and now is blue. So you've changed the genotyping call for that sample from a het to a home. The reason that this has happened is then highlighted by the red arrows. The analysis software that's out there for clustering for fluorescent probe-based PCR auto-scales the data. And so if you don't have enough raw data to create the different clusters, the software won't auto-scale the axes, and you will actually, unfortunately, misinterpret the data and potentially make an errant call. So because of the limitations of the algorithm, it really then does dictate how you design your experiments. So as a summary to the simple genotyping portion of my talk, as you can see, it's a very simple protocol. It's running just a standard PCR. You isolate DNA, create a reaction mixture, put it into your PCR instrument that's already available in your laboratory that performs high-resolution melting, and then you can collect data for as little as a single sample. So you don't have to batch data up till the end of the week. You don't have to worry about trying to combine data sets, and clustering algorithms are not going to fool your data interpretation. So this again yields HRM-based assays as how to make difficult experiments accessible to you in a laboratory. And so what I'm really trying to paint for you as a picture is that HRM-based assays allow you to address difficult targets, things that have difficult sequence, large deletions, heteroplasmic and mitochondrial DNA targets, copy number measurements, and all of this can be done in your own laboratory using a very simple protocol, very easy to do data analysis, and you can do it in as few or as many samples as you would like. Canon BioMedical has commercialized our expertise in HRM assay design into some products that are shown here, the Novallele genotyping assays that are available for either SNPs or insertions and deletions. We recommend using the associated Novallele genotyping master mix, which is a rapid chemistry. Generally, the protocol takes less than an hour for the run. These are shipped to you under environmental conditions, so it saves some money on shipping. And then you just store them in the refrigerator so that you can really set up your reactions as soon as quickly as you would like. If you already have designed HRM and would like to use our rapid chemistry, then you can essentially just convert your assay over to our chemistry using the 2x oligo dilution buffer. So with this, I've talked a lot about different biological conditions, and I've talked about the mechanics and technical information behind high-resolution melting. There are different ways to find our assays on our website. I've shown you two here. One on the left-hand side is if you're interested in a particular area of biology, you can browse through cancer or cardiovascular, immune disease, neuroscience, pharmacogenetics for listings of assays. Or you can use our search interface, and through the magic of the LabRoots virtual webinar, virtual presentation, we're now going to push you a link that's going to open up in your browser a new window or a new tab, and that's going to now open up the link for how to go ahead and search our website for your own target of interest. So you can choose those application areas. If you're interested in pharmacogenetics, look at that drop-down menu for drug. You can actually look for the variations that are associated with individual drugs. We have over 160 different drugs listed that you can look at the associated genetic variants, and that's all driven by the PharmGKB database. So we're going to push that link to you now so that it opens up in your web browser so that you can play with that. With that, I'm going to go ahead and wrap up here. Through my history presentation at the beginning of today's talk, I was able to talk about how Canon BioMedical is your new partner in biomedical research. We actively do look for collaborations with other companies, as evidenced by our work with T2 Biosystems and Spartan Biosciences. We're working with universities to develop interesting genotyping targets, and we've now launched that commercially to you as the research community in the form of the Novallele assays and chemistry that are designed for rapid PCR followed by HRM. We have a lot more products that are going to be coming out in the very, very near term, so I do hope you stay tuned for that. So with that, I am going to say thank you very much. I certainly appreciate you guys hanging on there with me through my getting over my cold, so thank you very much for that. We're going to go ahead and start taking questions, and then I'm going to go ahead and also ask my last poll question for today is that we're going to push out is, if you would like, we can go ahead and actively follow up with you about our solutions or provide additional information. So if you go ahead now and just go ahead and answer the poll that's coming up, if you'd like a Canon BioMedical representative to follow up with you to talk about your tough genotyping challenges and maybe how we could simplify it for you, please go ahead and say yes. We'd be happy to contact you and see if we could support you more directly. And with that, I also encourage you to open up the question and answer tab and go ahead and fill out whatever questions that you might have for me to go ahead and answer. With that, I'd like to say thanks.
Well, it's been an absolute pleasure. Just excellent presentation, Dr. McNally. Thanks for bringing that information to all of us. And as you already touched on, before we get started with all your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many of them as we can. And let's see, we already have a few questions queued up for you. The first one is, may I send samples to Canon for genotyping?
That's a great question. We get this a lot. Canon BioMedical really right now is focused on providing solutions that are simple to use in your laboratory. Canon BioMedical does not accept samples where we do it as a service. If you typically send your samples out to a core laboratory for genotyping, certainly you can encourage them to contact us, and we'd be happy to work with those core laboratories to provide that service. But Canon BioMedical does not offer genotyping as a service today.
Okay, that is good to know. Okay, the next question here is kind of a compound question, and it asks, what is the best sensitivity of HRM somatic mutation detection, and can it be reached? In other words, I mean, what % mutated DNA against WT background?
Yep, it's a great question. So there are published examples going below what we have published. But if you look at the heteroplasmic example, for mitochondria, that's really sort of a very similar experiment where we created admixtures diluting out a particular mutation. And in that particular case, the percent mutation that was detected was 13. But that was 13%. Now that's really, I would consider, an overestimate, though if you focus on somatic mutations, you can drive that percentage using HRM to something that's far lower, probably by about a factor of 5 to 10 in the published reports that I've read. So I hope that helps answer.
Excellent. That's great information. And there is actually another follow-up here. It says, what kind of DNA controls should we use for HRM, amplicons or plasmids or human DNA, or human DNA samples?
Yeah, I mean, I think most, most of the reviewers that are out there would certainly encourage you to use authentic human samples. However, from the perspective of the analysis, amplicons and plasmids, amplicons are a little bit better to work with. If you were going to do plasmids, I would say you'd want to linearize it. And then the only thing that I would encourage you to remember is that, excuse me, is the DNA that you want to put into the reaction should be in an equivalent buffer. So you don't want to, prep, for instance, DNA samples using different DNA isolation kits. So we just recommend that they are in equivalent buffers when you put it into the reaction. So first choice would be human DNA samples. Second choice would be amplicons.
Excellent, excellent. Okay. Our next question. With copies of mitochondrial genes also being present in the nuclear genome, how do you know that the variants you are detecting are not coming from the nuclear genome?
It's a great question. There are examples of the mitochondrial genes, mitochondrial genome having insertions in the human genome. The signal that we're detecting would be clearly above that detection limit. So, as an example, as I tried to point out during the course of the talk, there are anywhere between 2 and 10 copies of the mitochondrial genome in a cell, in a mitochondria, and then there's hundreds of copies of mitochondria in a cell, and that would easily sort of outnumber the 1 or 2 copies of a mitochondrial gene in the human genome. That, in combination with a robust bioinformatic-based design for the assays that would control for any sort of pseudogenes or specific or non-specific targets, are also being addressed. So that's why we're confident that the variants that I showed you in the publication are not resulting from a copy in the nuclear genome.
All right, thank you for that. And let's see, Dr. McNally, it looks like we have one final question, so if you'd like to answer this and then close out with any final comments, please do so. The last question here asks, how should I analyze HRM data?
That's a good question. So we've indicated that the HRM is really something that most fluorescent-based thermocyclers are compatible with. And in a lot of those instances, the instruments themselves come with an HRM analysis module. So we would encourage you to begin your analysis using the included software that you've essentially already purchased on your instrument. So you don't really have to go out and purchase additional software options.
Wonderful, wonderful. And with that, any closing comments or? Okay, it looks like we are good.
I enjoyed it. My side?
Yes, yes. Do you have anything else? Okay, well, let's see. Well, thank you so much once again, Dr. McNally. I'd like to thank our speaker, Dr. Brian McNally, as well as our sponsor, Canon BioMedical, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through February 2017. You will receive an email from us, LabRoots, alerting you when it is available for on-demand. and it will also be posted on labroots.com. You are welcome to forward this announcement to any of your colleagues who weren't able to join us today. Thanks for logging on and participating in today's broadcast. We'll see you next time here at LabRoots.